Hi everyone, welcome to my channel Pure Biology, and today come with another video, that is the shoot apical meristem. This is obviously very brief description about the shoot apical meristem, and in my previous video I have explained that the root apical meristem, that means how the tissues are involved in the formation of the root apical meristem. So here this is the brief description about the shoot apical meristem. Okay, and before going to this topic, I want to say that if you are the newcomers in my channel, so please you share and give a like for obviously this explanation, and because if you give a like, then I will be motivated, and if this is helpful to you, then you subscribe my channel. Okay, so here the shoot apical meristem is what means how this is developed. Okay, so in case of shoot apical meristem or the shoot apex, that consists of the meristem of two types of shoot apex, that consists of two types of region. What are that? One is the apical meristematic region and another is the leaf primordium. Okay, so shoot apex region that consists of two regions. One is the apical meristematic region and another is the leaf primordia. This is the lateral region, you can see. The short form of the shoot apical meristem, that is the SAM. That is SAM. Okay, SAM. So what is saying that this SAM is indeterminate in the development. Okay, this is very indeterminate in development. Here the two meristematic tissues are also seen. One is the vegetative meristem and another is the floral meristem. Okay, if the vegetative meristem cells that divide, they form all the tissues that related to the stem, and if it is converted to the floral meristematic tissue due to the division of that cells, that induce the apex region to form the flower. So in very induced flower, to here that is the vegetative meristematic region that is converted directly into the floral meristematic tissue. Okay, so this region or this tissue that is converted to floral meristematic tissue, and that's why the flower formed in the apical region. Now the floral meristems that are obviously determinate. Okay, and two different concepts arise here for the development of the shoot apical meristem. The first is that the layers. Okay, and the second is that zones. So the two concepts that arise for the description of the development of the shoot apical meristem. So in case of layers, the concept is that three layers are there. Okay, three layers are there. One is L1, another is L2, and third one is the L3. So L1, that is the outermost layer. Okay, this is the outermost layer, and here the cell divisions occur in the anticlinal plane, that means the vertical region. Okay, so here due to the division of the cells that produce the parts of the shoots. Okay, the parts of the shoots, or you can say the epidermis, form the epidermis. L1 cells, the cells that present in the L1 that divide to form the epidermis of some parts of the shoots. In case of L2, the cell divisions that seen in the periclinal and anticlinal plane, and here the divisions the cell divisions occur to produce some organs that are needed for the stem. And the last one is the L3. The cells divisions are restricted in both plane, obviously the periclinal and the anticlinal plane. So periclinal, you know the vertical, anticlinal, you know that the vertical plane. So periclinal is the horizontal plane. Clear? So due to the cell divisions of the L3, that produce what? That produce the lateral organ. Clear? So this is the concept of layers. And the second concept of these zones, that means scientists said that there are three zones. One is the central zone, second one is the peripheral zone, and third one is the rib zone. So the central zone, that is that mainly help in the formation of the organizing center. Clear? That helps in the formation of the organizing center. And the peripheral zone, that generally help in the formation of the leaf primordium. And the rib zone, that generally helps in the formation of the internal tissues of the stem. Clear? Now we have to learn about the central zone, peripheral zone and rib zone according to the book. What is that? The central zone, where it is located? This is located at the distal end of the apical meristem, and obviously it includes the all three layers that is already described here. The all three layers L1, L2 and L3, all three layers are present, or these are restricted within the central zone, and the cells divide less frequently and have prominent nucleus. So cell division is occurred here very less frequently, and the prominent nucleus present in every cell. The second, and you know that this central zone that produce the organizing center. The second one is the peripheral zone, that consists of the cytoplasmically dense cells. It consisting of the cytoplasmically dense cells, many cells, dense cells with proper cytoplasms, and these, the divisions of these cells that produce the lateral organ or the leaf primordium. And the rib zone, that lies where? Underneath the central zone, and it divisions of the cells in this rib zone that produce the internal tissues of the stem. So this is the concept related to the zones. Okay, and here the two important genes molecularly, another thing is explained by the scientists, that the two important genes are here involved in this formation of the shoot apical meristem. The one gene is that the shoot meristem less, or STM, and another gene is that WASCHEL, or WUSCHEL, or WUS. It is also known as the WASCHEL. So WUS. Two genes are involved also in the development of shoot apical meristem. So here in this video I don't try to explain about the role of these two genes. So I have to explain because I have to explain about these two genes in the next video. Why? Because this is the another topic related to this SAM, but this is another topic related to the molecular biology. Okay, so I have to explain that in my next video. So if you interested in my channel, so continue with my channel for the next video. So thank you.